it's now time to recognise and hear from some of Australia's most outstanding researchers and to get on with the Academy's annual awards. And I must say, it's particularly wonderful to have here amongst us some of the donors whose continuing generosity allows us to make these awards. As you all know, they recognise scientific research of the highest order, and it's always a privilege to hear from the recipients. We'll begin today with addresses from our career awardees, and then we'll follow on by hearing from our early and mid-career awardees. And to help present the awards and chair the session, I'll shortly hand over to the Academy's Secretary for Physical Sciences, Professor Jagadish, and the Secretary for Biological Sciences, Professor Marilyn Renfree. But first, I have the pleasure of introducing the winner of this year's Macfarlane Burnet Medal and Lecture, which recognises scientific research of the highest standing in the biological sciences. This award commemorates the enormous contributions to science by Foundation Fellow and former Academy President and former Director of the Walter and Eliza Hall Institute of Medical Research, the late Sir Frank Macfarlane Burnet. The medal will be formally presented at the dinner tonight, but we'll hear the lecture this morning. So I'm pleased and very proud to welcome to the stage the recipient of the 2014 Macfarlane Burnet Medal, Academy Fellow, Professor Gerry Adams.
Well, it goes without saying that I'm delighted to receive this.
Professor Adams has advanced the understanding of cancer development, particularly of genes activated by chromosome translocation in lymphomas, through molecular analysis and transgenic mouse models. By clarifying how the BCL2 protein family controls the life and death of cells, he and his colleagues have galvanised the development of a promising new class of anti-cancer drugs that directly engage these death regulators. Congratulations, Professor Adams. I now invite you to present the Macfarlane Burnet Lecture entitled Control of Cell Suicide by the BCL2 Family.
Thanks very much, Marilyn. Sorry about jumping the gun there. As I started to say before, of course, I'm delighted to receive this, but also humbled by this, both because I realize how many talented colleagues contributed to my own work, but also because how many worthy recipients there would be in this fellowship. And of course, it's rather special having anything named after Burnet, because he was obviously one of the most outstanding Australian biological scientists. And he's, of course, a real hero in our institute because his wonderful discoveries in first virology and then immunology thrust our institute onto the international stage. With that having been said, I must admit that I'm rather glad that Burnet was not around to be on the selection committee, because he had a rather negative attitude about molecular biology, which he once described as largely a laboratory artifact that has never been brought into useful relation with biological realities. And he thought also, you can see down below, that potential for cancer research looked pretty hopeless. So my subject of cell suicide might seem like a rather grim topic, that only is relevant to pathological situations. But in fact, cell suicide is vital for normal physiology. It sculpts embryonic tissues, as shown most obviously externally by the removal of the webbing between our digits, except for creatures like a duck would keep it. And it has much more vital roles internally. It's critical for proper immune function. It culls infected cells to limit pathogen spread. And perhaps most important, throughout life, it ensures tissue homeostasis by balancing cell proliferation. In organs like the gut, liver, or the hematopoietic system, where you have robust cell proliferation, if we didn't have cell death, we would all explode. So, it's a vital function, a vital process, and we now appreciate that too little cell death can promote cancer and autoimmune diseases, and the latter if it happens in lymphocytes. On the contrary, too much cell death can favor degenerative disorders, perhaps including things like Alzheimer's disease. So for these reasons, all of our cells are in fact poised to commit suicide. That hasn't, however, been known forever, and it was in fact John Kerr, a Queenslander, who first noticed the orderly morphological changes that often occurred during physiological cell death. And he argued that he and colleagues, two UK colleagues, Wiley and Curry, argued in a seminal review in 1972 that this meant that it had to be programmed. Cell death wasn't accidental, it was in the main something that happened because cells had determined to die. And the argument was that for this orderly progression was that cells would shrink, and then the chromatin around the nucleus would condense, and then the cell would become segmented into membrane-bound bodies called apoptotic bodies, which were then consumed by neighboring cells. So the whole process is often almost invisible in tissues, even with a lot of apoptosis going on. So it allows the tidy removal, and by the way, they in fact coined this term apoptosis. Apoptosis allows tidy removal of redundant or dangerous cells, precluding release of their inflammatory contents. But there was no molecular mechanisms. It took another 16 years before the first molecular player, could be identified. And that was made possible by work in several laboratories, not ours, that revealed a chromosome translocation that involved the immunoglobulin heavy chain locus and a previously unknown gene, which they dubbed BCL2, because it was the second B-cell lymphoma-associated gene that they had studied. We immediately recognized, because we had earlier done work where we had identified a chromosome translocation where the MYC gene was brought into close contact with the immunoglobulin heavy chain locus. And we knew that in this situation, what actually happens is that the gene has been brought close to a potent enhancer, transcriptional enhancer, that means that actually now BCL2 would be made in much greater amounts than normal in lymphocytes because that enhancer works very efficiently in there. But having found this gene didn't actually immediately tell much of anything because it wasn't at all clear what it might do. And that was only finally resolved by a seminal study done by David Vo while he was a PhD student with me. Great way to launch your career. Because he found that BCL2 prevents the death of hematopoietic cells when they've been deprived of the cytokines that they normally need to multiply. So you see in experiments shown here, the cells that were forced to express BCL2 by a retrovirus survived when they were deprived of cytokine, whereas all those that weren't infected died very quickly. But they did not proliferate. And David went on to show that BCL2 cooperated with the MYC gene in cell immortalization, another step towards tumor development. So these studies showed that programmed cell death, or apoptosis, is regulated separately from cell proliferation and that its impairment is very likely to promote tumorigenesis. And that was confirmed and extended by in vivo studies done here by a very young Andreas Strasser under the tutelage of Suzanne Corey, using so-called transgenic mice, that is, mice which we had constructed, using that enhancer region and connecting it to either BCL2 or much earlier to the MYC gene. And so what you're seeing here is the cumulative mortality in mice that carry either BCL2 transgene, a MYC transgene, or both transgene. And it's obvious that BCL2 promoted tumor development, at least in the context of a MYC transgene. And we now understand that pretty well because MYC not only promotes proliferation, but if it promotes too much proliferation, it drives the cell through cell cycle checkpoints in an unwarranted way, and that leads, in fact, to apoptosis. So BCL2 actually counters the apoptosis driven by deregulated MYC expression. So together, these two studies promoted the concepts that impaired apoptosis is a critical step in tumorigenesis and probably also a barrier to durable cancer therapy, because it turns out that BCL2 promotes survival in the face of all sorts of cytotoxic signals. Of course, BCL2 doesn't work alone, and I'll now skip the history and get to what we know now. It turns out that this family is a rather remarkable one, involving 3 different factions, in which 2 of the factions, so-called BH3-only proteins, which I'll explain a little later, and BAX actually promote cell death, whereas BCL2 and its closest relatives promote cell survival. So it's sort of a contest inside this family based on molecular interactions that end up determining whether a cell will live or die. And to give you a little bit more background, the BH3-only proteins are essentially signaling molecules, have very little structure, except for the BH3 domain. That name comes from being one of the regions that's similar within the family. So they only have one similarity to the rest of the family. And they work by binding the pro-survival proteins, and some can actually directly attack also BAX, it turns out. The pro-survival BCL2 group work by keeping BAX in check until they are overwhelmed by the BH3-only proteins. BAX are the real drivers of apoptosis because they form oligomers that damage the mitochondrial outer membrane where most of these interactions occur. And the reason that's so critical is because when they damage that membrane, cytochrome C is released from the intermembrane space of the mitochondrion, and it actually serves as a cofactor that elicits the proteolytic demolition of the cell by a dedicated group of proteases called caspases. It's also very important to note the remarkable similarity between BCL2 and BAX. They're very similar globular bundles of 9 alpha helices, where the last one is an anchor to the mitochondrial outer membrane. And it's only in the last year or so that we've come to understand the fundamental differences between BCL2, pro-survival, BAX, pro-apoptosis. So the family relies upon interactions. And the canonical interaction of the family is the binding of a so-called BH3 helix, which I'll explain now, into a groove on the surface of the globular relatives within the family. And you can see that shown here, where the yellow indicates this helix bound into this groove on BCLXL, which is very similar to BCL2. Now, what is the BH3 alpha helix? It's about 20 amino acids, but there are about 5 or 6 residues that are particularly important. 4 hydrophobic residues, sort of greasy residues, protrude into greasy pockets on the protein. whereas one acidic group, acidic aspartate, forms a salt bridge with a basic arginine in the protein. So that's essentially what a BH3 is. And the binding of a BH3 domain inactivates pro-survival function, probably by preventing the pro-survival protein from binding to the BH3 of Bax or back. So that's the central way the family works. It's interesting that we found out that there is some specificity in the associations, and it's rather interesting, and it's relevant to the drugs that have been developed to attack this family. That is that not all the BH3-only proteins react with all the pro-survival proteins. In fact, the most potent ones, the ones that kill best, do interact with all the known pro-survival proteins. But the ones that bind more selectively to a subset of the proteins are also weaker. However, if you combine together two of these weaker BH3-only proteins, you get efficient killing. So this suggests that efficient apoptosis requires neutralization of all the pro-survival proteins expressed in a given cell. And that's very relevant to the drug possibilities that I'll now discuss. The drugs that attack the pro-survival members of the family are often called BH3 mimetics because they mimic the function of the BH3 domains by inactivating the pro-survival function. And of course, you'll be not surprised to learn that they do that by binding into this groove in a manner somewhat like the real natural BH3-only proteins. So what's the rationale for this kind of therapy? Firstly, tumor cells often resist apoptosis by defective induction of BH3-only proteins. For example, if you knock out as often happens in about half of our tumors that develop in humans, a mutation in the p53 gene, you get very poor induction of BH3-only proteins. And as I mentioned, some other tumors have excessive expression of a BCL2 pro-survival protein. But the core apoptotic switch remains intact. And therefore, the idea is that the BH3 mimetics drugs, if they're high enough affinity, should overwhelm the pro-survival proteins and induce apoptosis. And in fact, that looks to be the case. The first authentic BH3 mimetics were developed by Abbott's laboratory. And the classic one was ABT737, but they also developed a clinical derivative, which is now called navitoclax. And they hit 3 of the pro-survival proteins. And in fact, here's this picture done by our structural biology colleagues, shows in fact ABT737 binding into that groove. Obviously, it doesn't at all really look like a peptide binding there, but it hits some of the same pockets and therefore has high enough affinity to do the job. We showed that these drugs work best on cells with high BCL2 and low MCL1 and A1, as you might suspect from the specificity. As single agents, they kill some tumor types, particularly lymphoid ones, and the killing of many more if they're combined with conventional cytotoxic agents. First clinical trial of the navitoclax was performed in Melbourne and led by Andrew Roberts, a OEI colleague. And that study pleasingly showed that about 35% of patients showed some benefit from the treatment. However, very high doses weren't able to be used because there was killing of platelets by the BCL-XL inhibition. So it was entirely an on-target effect, but it was unfortunate because it was killing too many platelets for it to be safe to go up in the dose, suggesting we need to have more specific drugs. And so to develop new BH3 mimetics, our institute collaborated with Genentech and Abbott And our collaboration helped produce the first BCL2 specific agent. The majority of the work actually was done by Abbott in creating this compound ABT-199, which is highly specific for BCL2. And therefore, the amount of platelet killing was 100-fold lower than it was with navitoclax. And that is held up in patients, where the first few chronic lymphocytic leukemia patients have had a marked fall in tumor cells in the blood, but not in platelets. So ABT-199 appears very promising, particularly for lymphoid malignancies, and it will probably have a wider efficacy together with other agents. So the hunt's now on for other molecules that will target other family members, such as MCL1, and our institute is again participating in that search. Now I want to turn and talk to what I consider the most poorly understood aspect of apoptosis, and the one that has fascinated me for the last few years. And that's the question of how BACs and BACs are converted from inert monomers that do nothing to lethal oligomers. And the first clues about this came mainly from two of my colleagues, Ruth Cluck and Grant Dewson. And we proposed, firstly, that an activator BH3-only protein, such as BIM, induces BAC to expose its own BH3 domain, which is actually the alpha-2 helix. And I've tried to indicate this schematically here, that the BH3 of BAC is initially sort of buried, and it flips out, and it becomes exposed. And then we suggested that two of these molecules get together to form a so-called symmetric dimer. And then we proposed that these dimers multimerize by another link, perhaps involving alpha-6, to form the pores that trigger cytochrome C release. And to get at this, we obviously needed to have structural information. And so we entered into collaboration with our structural biology colleagues at WEI. And Peter Coleman, I think many of you will know, a fellow of the Academy, had a key role, as did his talented junior colleague, Peter Zabitar. And our work with them led to the first structure of BACs with a bound BH3 peptide. And you can immediately see that the binding is remarkably similar to that, to the pro-survival proteins. It involves these four hydrophobic residues and the salt bridge. But there was, fortunately, an extra pocket in BACs, and that told us a bit more about why BID, BH3, is in fact an activator of BACs, but not some of the others. The key thing, however, the remarkable thing about the interaction with BACs is it actually changes the structure, and I show that here. What we think happens is when BH3 binds to BACs effectively, it actually unfolds, maintaining a core and what we call a latch that pulls away from it. The core domain seems to be essential for oligomerization, and therefore we believe that it will, the core oligomerizes and carries along the rest of the molecule. I believe I'm out of time. So I'll say thank you.
Thank you very much, Professor Adams. Questions? Nick? Wait for the microphone, Nick. Please. No, it's because it's being recorded.
What is it that triggers a cell to go into apoptosis? What is the tipping point?
It can be all sorts of cytotoxic signals. They all seem to converge on the BCL2 family, and they're all fundled by these BH3-only proteins. And so there are many ways in which the BH3-only proteins are activated. They can either be by transcriptional or post-transcriptional alterations. So basically, they then carry the signal, and they interact with the pro-survival proteins, and when enough of this kind of signal builds up, when it inactivates enough of the pro-survival proteins, then backs and back, go ahead, oligomerize, and kill the cell.
Jerry, I'd like to ask you about those human conditions where you have webbing, persistent webbing between the digits. Are they because of a failure of the BCL2 proteins?
That's right. If you have a knockout of certain critical pro-apoptotic genes, such as backs and back, you maintain the webbing. And you also cause much more serious disruptions to the neuronal system, so that the mice die near birth.
So how is it regulated in aquatic species where the webbing is retained?
That's not really known. Not too much studies have been done outside of mice and humans and there's some invertebrates, but the control mechanisms there are rather different.
Jamie.
Two questions. Firstly, the conservation of this process throughout evolution, and secondary, is then the promotion of oligomerization a therapeutic avenue as well?
Let me answer the first question first. The evolutionary conservation is there, but it's a little bit complicated. The basic idea that of turning on an apoptotic process is definitely conserved. And for example, the best studied organisms are C. elegans and Drosophila, as far as cell death is concerned. In C. elegans, you have a BCL2 homologue. You have a molecule that's similar to the BH3-only proteins. It has a similar function, but you don't, in fact, have a BAX analog. And so it looks like the mitochondrial disruption is not the way it works in C. elegans. Instead, it works by direct contact between the pro-survival protein and a regulator of caspases. And it's even a little bit more obscure what happens in Drosophila, where there are a couple of BCL2-like molecules, but they don't seem to be the really critical determinants of cell death in Drosophila.
Fred.
Fred Mendelsohn. Why is it, Jerry, that these agents don't trigger apoptosis in normal cells? Why are they relatively tumour-specific?
That's a great question, Fred. First of all, we think a lot of it hangs on which pro-survival proteins they attack. As I mentioned to you that even the first navitoclax only attacks 3 of the family members. It boils down to saying that the tumor cells seem to be often sort of primed for death. And that seems to happen probably because their apoptosis has been compromised, and that leads to the buildup of complexes of the pro-survival proteins with excess amounts of BH3-only proteins. So the drugs probably actually work in part by releasing this flux of excess BH3-only proteins, and that's probably what ends up killing the cell most, the tumor cell, more than it does the normal cells. But it is a fine balance, and how far this approach will go will require a lot more study, because there will always be effects on normal cells.
I'm afraid we don't have time for any more questions. I'm sorry, Eugenie. So would you please join me in thanking Professor Adams for a fascinating talk.